Today, my GD topic is classification of crops based on CO2. Classification of crops based on carbon dioxide is one of the basic work, one of the basic element for the plants in the process of respiration. We know well. We know the process of respiration in which plant, while doing the photosynthesis, plants need to breathe through stomata. So they do respiration process in which they inhale oxygen from the atmosphere and exhales the carbon dioxide. Well, we know this process. This process occurs generally in the daytime, but in the night time, it got reversed. In night time, they inhale oxygen and exhale carbon dioxide. And the reduction of the carbon dioxide is also called the dark reaction, as light is not necessary. And the dark reaction does not mean that it occurs only at the night time. In few plants, it also occurs in the daytime. While this process is generally work for the energy supply by the ATP, which is producing basic form is to produce the carbohydrates to supply to the plant. Okay, and we know that NADPH, which was also supplied while CO2 combines with hydrogen, which is also known, the full form of the NADPH stands for nicotinamide adenine dinucleotide phosphate. Well, this particular thing get combines with hydrogen and CO2, so they form carbohydrates. By the method of CO2 reduction, plants get classified into following categories. The first category is C3 plants. The C3 stands for 3 carbon. While the 3 carbon, while the C3 plants are the normal plants, and there are 85% of the plants species known as C3 plants, including white, reed, soybeans, and all the trees. So the first step of the Kelvin cycle is the fixation of the carbon dioxide, the rubisco. Rubisco works with 6 carbon, but here in C3 we are having 3 carbon. And the plant that usually only standard mechanism to carbon fixation are called C3 plants for the 3 carbon only. So when the CO2 enters through the stomata, it meet with rubisco and form the 3PGA, the pair. In that it get involved in the Kelvin cycle. From the Kelvin cycles, they produce carbohydrates, basically sugar. Okay. The next category is C4 plants. C4 for the 4 carbon plants. As we know, in the C4 plants, the light dependent reactions have the Kelvin cycle are physically separated, with the light dependent reactions occurring in the mesophyll cell, the spongy tissue in the middle of the leaf. After when you are seeing the leaf through the microscope, we see so many pores and the stomata. There is a spongy part of the leaf which is in between the layers. So when the CO2 enters, it get meet with the PEP carbolase. PEP carbolase, we know PEP is phosphophenol pyruvate carbolase. Then it, when they meet, they form oxaloacetate, then to mannate. From there, if the CO2 get divided, it met rubisco. Then the step of the C3 photosynthesis will occur in the Kelvin cycle. But if not, then they will form pyruvate, then PP. When the CO2 gets separated, it will form pyruvate, then it will form PP, phosphophenol pyruvate. Okay. So basically, inside the bundle set, the mallet breaks down, releasing a molecule of CO2. The CO2 is then fixed by the rubisco and made into sugar by the Kelvin cycle, like we have studied in the C3. Okay. Now we'll know one of the major steps, that is the step is carried out by the non-rubisco enzyme, the PEP carbolase, that has no tendency to bind O2 oxaloacetate. So the rubisco is one of the major material that can bind with anything. It will bind with O2, it can bind with CO2. So then it converted to a similar molecule malate, as we are seeing oxalated then to malate. Then it will, CO2 will breaks down and will meet the rubisco, fix with rubisco and make the sugar. Now the third one is the carcinosin acid metabolism. The camp plants. Camp plants, the general thing about the camp plants, that they are grown in a very hot and dry areas like the deserts. For example, we'll see the cactus. The basic form of the cactus is that when in the desert there are such a high temperatures going on, and then the cactus can't take that energy while having so much temperature. The direct sunlight is making on it. So the photosynthesis process, the cactus generally occur in the night time. Some of the plants that are adapted to the dry environment, such as cacti and the pineapples, use camp pathway to minimize the photorespiration method. So as in the diagram we can say that starts to triphosphate to phosphonyl pyruvate PEP, then to HCO3 PC, then to oxidatate, then the whole process of the C4 and C3 are temporarily separated in this process in CAM plants. Okay, they then the main thing is that instead of separating the light dependent reaction, use of the CO2 in the Kelvin cycle in space. CAM plants separated this process in time. At night, CAM plants open their stomata, allowing CO2 to diffuse into their leaves. So here we are seeing that the light dependent reaction of the plant of the CO2 is a Kelvin cycle in the space. So the CAM plants are separating this process. It is separating all the process in that time. But in the night the CAM plants open their stomata. So they are allowing CO2 to diffuse into their leaves. This CO2 is fixed into the oxalated by the PEP carbonase. The same step used by the C4 plants, like the C4 process, then converted to a malate to another type of the organic acid. The organic acid is stored inside the vacuoles until the next day. In the next day, like the CAM plants do not open their stomata, but they can still photosynthesize. That's because the organic acids are transported out of the vacuole and break down into the release CO2, which enters the Kelvin cycle. So this controlled release maintains a high concentration of CO2 around Rubisco. The CAM pathway requires ATP at the multiple steps. So like the C4 photosynthesis, that's not energetic freebie. Thank you for your patience listening and have a nice day to you.